Assalamualaikum and hi everyone. Good morning to Dr. Prasita and fellow friends. So today I am Rashkin and my partner Rafia Rashma will explain about one of signaling pathway called as Wnt Signaling Pathway. The rest is our team members. So next, what is a Wnt Signaling Pathway? Okay, Wnt Signaling Pathway is a gene of the growth stimulatory factor. In the mammals have 19 Wnt gene. So the other name for Wnt signaling pathway called as beta-catenin, which is regulated by degradation. Wnt signaling is highly conserved and is very important and have a vital role in embryonic development, such as for proliferation and also cell movement. There are consist two transmembrane receptors. The first one is Frizzle and LRP, which is a lipoprotein. So, in Wnt signaling pathway, there are three pathways involved. Two of it is involved inside the normal stem cell, which is normal and oncogenes. What happened to the third one? The third one involved in cancer stem cell. Is known as tumor progression or invasive cancer. So let's go through one by one. So the first one is a normal pathway. Normal pathway of Wnt signaling is the Wnt signaling is absent. So the protein beta-catenin bound by the destruction complex. What is the destruction complex? The destruction complex are CK1, APC, exin and GSK3 beta. Okay. Then which include protein kinase. The protein kinase phosphorylated beta-catenin targeting it to ubiquitination and degradation. Who will degrade this? The degradation will degrade by proteasome. Okay, the beta-catenin will degrade by proteasome. There is no beta-catenin free to move into the cell. So what happened to the nucleus? The transcriptional co-repressor bind to the TSC function, which is prevent the expression for catenin genes. So next, I will pass to my partner to continue on oncogenes and also tumor progression.
Okay, in the oncogenes will be present of the Wnt signal. The first step, by binding of the extracellular factor Wnt to the membrane receptor called Frizzle and to its co-receptor called LRP, the Wnt pathway is activated. Binding leads to the activation of the cytosol protein disheveled abbreviated DVL. DVL induces dissociation of the distraction complex. Because beta-catenin is no longer modified by ubiquit peptide, so it is not destroyed. Since the supply of beta-catenin is continuous, the level of beta-catenin will rise, first in the cytosol and later also in the nucleus. In the nucleus, beta-catenin together with other protein binds to the TCF-left transcription factor, which changes from a transcriptional repressor into an activator. Then, TCF-left itself activates an RNA polymerase, which induces gene transcription. And next is tumor cells. Next slide. In the tumor progression, there are no presence of the Wnt signal. In the tumor cells, the Wnt pathway may be activated in the absence of the Wnt signal. Most often, a gene that codes for a protein of the destruction complex carries a mutation. As a consequence, the complex disintegrates and can no longer take beta-catenin for distraction. So the level of beta-catenin will also increase here. This situation reflects the situation in a oncogenes normal cell when a pathway is activated by a Wnt factor. In the nucleus, beta-catenin activates the TCF-left factor, just like in the normal cells, after activation by Wnt factor, TCF-left also activate an RNA polymerase, which starts transcription of several genes. So that's all from us, and thank you.